first of all, I know there's no greater honor than being asked to distract people while they're drinking. So thanks to all of you, and thanks to the Pint of Science for organizing this. And I'm going to talk about how bacteria become multi-drug resistant, unkillable superbugs. But, so this event is called Pint of Science. And there's actually a popular misconception that science is how the world works. No, the world is how the world works. Science is just a bunch of bumbling hominids all working together trying to figure out how the world works. So I'm going to tell kind of a little story about how I've come across what I've found. So first of all, who am I? What kind of hominid am I that I get to call myself a scientist? So I'm a postdoc. What does that mean? Well, you can think about academia, at least, so science in a university research lab, as one of the last bastions of medieval feudalism. So if you're an undergrad and you happen to work in a science lab at a university, you're kind of like a landless serf, a peasant, who does all the grunt work that's really important, but nobody else wants to do. Then if you decide to go to graduate school and get a PhD, you become a grad student. And that's essentially an indentured servant. So you've signed away 5 to 8 years of your life, and hopefully, eventually, you will earn your freedom dues in exchange for a 100-page thesis that no one will ever read. So then if you're not scared off by that, you can go off and become a traveling itinerant apprentice. So that's the postdoc, where you have your PhD, but you still need to learn a little bit more and kind of hone your craft. And then above them are the various ranks of full and assistant and associate professors who are like the landed nobility, the earls and the dukes and the princes who own their own land, who run the labs, and actually get to decide what science research gets done. So when I came here as an itinerant traveling apprentice and joined the lab, I was lucky enough that my Duke allowed me to decide what projects I wanted to work on. So the particular thing that I was really interested in was lung health and air quality, and then there's also this climate change thing that seems to be kind of a big deal. So knowing biology and bioengineering, I thought a really good way to try to tackle all of those at the same time would be biofuels. So I thought maybe I could come up with some way to make cheaper, more effective, more efficient, cleaner burning, low carbon, sustainable biofuels. So I thought really, really hard, and I came up with this great intricate idea for how you could evolve algae to make biofuels more efficiently. And so I tried to do that, and on the first try it worked, so I fixed everything and we're done.
Woo!
Wait,
Great speech.
How much time did it? Okay, so just kidding. Actually, it ended up taking a lot longer than I thought it would. I was making some progress, I think, but it was taking a long time. And I really only had so much time to try to prove that I could earn my keep. Because remember, my Duke has to fight really hard for limited research grants to fund his lab and keep it going. So he can't fund me bumbling around forever without coming to a conclusion at some point. So I decided I need to work on something, find maybe a side project that would be maybe a little bit easier, a little bit quicker to get something to just get it out and have it done. And so that's when I came across this paper about sword-fighting bacteria. So we've known for a really long time that bacteria fight each other with essentially chemical warfare. So they make chemical weapons, and we actually make those chemical weapons. We call them antibiotics. But with bacteria, they make them and just spew them out in the environment and try to kill each other with them. This sword-fighting mechanism, though, is really different because with antibiotics, they can just blindly spew them out and just hope that some of them will land on their enemies and get rid of them. But with sword fighting, they have to actually be physically touching. They have to be close enough that they can reach out with what is essentially a poison-tipped dagger and stab their neighboring bacteria and inject toxins into them, which will kill them. And that's interesting because that means all of a sudden now, spatial patterns matter a whole lot. So we've learned a lot about bacteria over the past several, well, 100 years-ish by studying usually one species at a time in a well-mixed shaking test tube with no spatial patterns because it's just shaking and it's well-mixed. But in reality, say in an infection in your arm, there's a real spatial environment there. And it's not just one species at a time, it's a lot of species all mixed together and interacting in weird ways, which apparently includes this sword fighting thing. So I thought about that for a while, and I was like, that's really interesting, because what this means is that maybe the bacteria are kind of playing a game of Risk. Right? So if you've played Risk, you know that you can only attack neighboring countries or territories or whatever these are. So if you take all your troops and you just spread them out across Asia, right, and then you engage in a land war in the winter, you're going to lose. But you could take that same number of troops and you could shove them all down here in Australia, and now there's only one way in. People can only attack you on one side because of the spatial geometry here. So you can just pump out armies, and it doesn't matter if everybody else out here is really stronger than you. You can just keep pumping out armies, and they just can't get in to get you because you're protected by sort of the spatial structure, the geography. So I thought maybe I could see this happening under the microscope and understand a little bit more about how bacteria are interacting in real environments like inside a wound. So I ordered one of these sword-fighting bacteria. I got that. So I'm growing this guy in the lab now. And I mixed it together with a poor, defenseless, docile, domesticated lab strain of bacteria that doesn't have this sword. And my idea was I should be able to see, like, just very first test, make sure that this actually works as advertised, right? So I want to mix them together and see if the red guy actually does kill the gray guy. So how can I test that? Well, what I do is I gave them different antibiotic resistance genes. So this red guy gets the resistance gene for antibiotic S. These are, I'm sure, going to fall on the floor. The gray guy gets resistance to antibiotic K. So I can mix them together in a petri dish and let them grow in this spatial environment that's not shaking. Oh, sure. So I'll put it on my incubator here. Are you 37 degrees Celsius?
Unless
Okay.
I have running fever, but I don't think so.
Okay. Well, we'll find out if it doesn't work. All right, so we're going to mix them over there. And now what I'm going to do is I have two more antibiotics. So you guys play nice. I have two more petri dishes here that have different antibiotics in them, S and K. So now I can gather them up after they're done playing and spread them on these new dishes, and only the red guy should grow on this dish, and only the gray guy should grow on this dish. So I can count how many grow on each dish, and I can make sure that, yes, the gray guy really gets killed off. So I did that, and of course, after mixing them together, I get a lot fewer cells that are growing on this K dish. So that's on a Friday, then I go home for the weekend and plan what I'm going to do next. And of course, because I'm a slob, I didn't throw away my dishes, and I came back in on Monday, and I noticed that on this K dish, where only the gray guy should be growing, the cells that are growing on it actually look red. So how does that happen? Because these red guys should not be growing. They have the S resistance gene. They should not be able to grow on this dish with the K antibiotic. Does anybody have any guesses what might be happening? Maybe. Yeah, so it must be that this K, somehow while they were playing together over here, this red guy picked up the K gene, and now it must be resistant to both of them. So that's what I thought must be happening. Now, we could test this, maybe. So we want to make sure that our theory here is correct. We could test it by making a third dish that has both antibiotics. And if we're right, we should get cells growing on here, but only after they have time to play together. So this is the experiment we want to do. This is the money experiment to prove our hypothesis, but there's a catch. And the catch is that in the meantime, I got assigned to a different project that the lab actually has dedicated funding for. Remember that? I said this was kind of my side project that I was interested in. So what are we going to do now? Well, luckily, an email came across my inbox from some listserv that I had somehow gotten subscribed to, and luckily I read it, and it was an announcement for this fellowship for postdoc funding. So I've actually written a few few fellowship applications before, and I've never gotten anything. So I've never gotten anything. I assume, obviously, it must be because true genius is never recognized in its own time. But regardless, I didn't have super high hopes, but this looked like it would be a good fit. So I wrote an application, I sent it in, and then I waited, and a few weeks later, I got this phone call from an unlisted phone number. So I picked it up and said, hello. Somebody says, your package came in. I said, my package? Fellowship package? Cool, what package? Cocaine. Wrong number? Okay, so I'm not answering my phone anymore if the caller ID is blocked. I've learned that lesson. A few days later, I get another phone call with the caller ID blocked. I'm not going to answer that. But they leave a message. So, okay, that's safe enough. I can check my voice. I listen to the voicemail, and it says, hello, this is the UCSD grants office. No, call back. So, okay, I call back, and yeah, I got really, really lucky, and I got this fellowship funding, which means that now I can chase down the project that I want to do for about 2 years-ish. So, we go back to this experiment. We have this 2-antibiotic dish, and we let the cells grow together for a while, and then we spread them on here. And of course, what do you think happens? Does anybody grow? Yeah, so this red guy now can grow, but only after we've grown it, mixed it together with the gray guy. Now, we can test this even more by, say, deleting this poison tip dagger gene. So we take that away from the red guy, and we let them grow together. And now does anybody grow this S plus K 2 antibiotic dish? Well, actually a few of them do, because nothing's ever clean. But it's much fewer than if they have this killing mechanism. Or the other way around, we could give kind of a suit of armor to this gray guy, give it an immunity gene so it can't get killed, and again, we get a lot fewer of this K gene transferring over to this red guy. So this seems really cool. So what I think is actually happening is that it's not just that they're playing Risk, but they're playing Final Fantasy. So because in Final Fantasy, in games like this, when you kill your enemies, they drop goodies that you can pick up and then gather in your bag or whatever and use for yourself. So is this really relevant out in the real world? Well, this red guy is known as Acinetobacter, and it's actually a really big problem in hospitals, not because it's like super pathogenic and super dangerous, super virulent, but in hospitals you have a lot of people with compromised immune systems who can get infected by cells that, by bacteria that aren't really a problem for healthy people. And because these guys in particular are really good at gathering lots of different antibiotic resistance genes, they become really hard to treat in these patients when they do get infected by them. And in fact, they get, these guys in particular gather antibiotic resistance really, really fast, much faster than people really have been able to explain before. So I think that this mechanism, this sort of sword-fighting Risk/Final Fantasy thing, where you kill your neighboring bacteria, that releases the genes that they were carrying, and then you can gather those up. That helps them to shortcut evolution, because they let the other cells do the hard work of evolving this gene, and then they just come in, kill them, and steal it. So, I think that's about all I had. That was my spiel. That's my little story about a hominid doing science, and a little bit about, I think, possibly one mechanism for why Acinetobacter and bacteria like this are becoming so multi-drug resistant. And I'll just summarize it up really quickly. I can remember this. Okay, so antibiotics. They were a health revolution, but not a total bacterial solution, because they sparked evolution of resistance genes that spread like pollution, especially in Acinetobacter, which evolves with a whoosh. And we don't know just how they acquire resistance with such speed and persistence, but they can kill their neighbors, the fiends, and that helps them steal genes, and they can even watch live under the microscope, in microscope scenes. But of course, you only know half it unless you can math it. So I wrote some equations, which I forgot to mention, and with pipettes and persistence and patience, I fit curves that match with precision the gene acquisition of these cells in addition. Eventually, I'd hope to help prevent or reverse antibiotic resistance before it gets worse, with tools and techniques for doctors and nurse. So, thanks to you all for lending your ears. Let me know if you have any questions, and cheers.
Okay, you can ask questions only if they rhyme. No, I'm kidding.
How many you don't make me answer it?
Yes, okay. Question.
Fart.
So you talked about developing tools to stop them from acquiring new genes. Can you give us some clues? Does it have to do with taking away their swords or anything else?
So of course, because I've chatted with you about this several times, I think you're getting at the idea of if these guys will pick up genes from the environment, they don't really have any way of knowing what genes they're picking up. They just pick it up, and it looks like they know what they're picking up, because if they pick up something bad, then they die and we don't see them. But if they happen to pick up a good thing, then that one cell they picked up the good thing spreads, and it looks like they all picked it up. So that's sort of the way evolution works, is it's random chance, and then whatever works is what you see. But on a shorter time scale, maybe if they're just picking up genes at random, maybe we could throw in like a Trojan horse gene and convince them to pick it up and figure out some way to spread that Trojan horse gene through the population so that maybe they would not be able to, so to get rid of their antibiotic resistance or to give them some new susceptibility. And I've been kind of working on that, and I've figured out many, many ways for it not to work. But it keeps not working in a different way, so I feel like I'm making progress. And eventually, maybe it will run out of ways not to work. But I'll keep you updated.
Have you looked at human isolates to see if there are specific genes that it particularly likes versus not?
So I have not, but actually the species that I'm working with, so the question was, what about, so I'm working with some lab species of Acinetobacter, but what about the ones that are actually found out in the wild in real patients? Is there some pattern to what resistance genes those pick up? And so the guy that I'm working with is actually Acinetobacter bailey, which is sort of a semi-neutered, safe-ish, safe lab strain that I can work with without having to wear a bunny suit, because those are uncomfortable and it gets hot here in the summer. But the problem in hospitals is Acinetobacter baumannii, and people have looked at those, and yeah, it turns out that the resistance genes, a lot of them that they have, so I don't know if a lot of you guys know this, but we've gotten really, really good, like astonishingly fast at reading DNA. So the Human Genome Project, if you remember hearing about that, it took like a decade or something to read the first human genome. We can do that now for like $1,000 in 10 minutes. So like way faster than Moore's Law in computing. So that means we can take bacteria that we find from patients and we can see what genes they actually have really quickly. And so a lot of these Acinetobacter guys, their resistance genes look a lot like resistance genes that are found in other pathogens that infect humans. So yeah, it does kind of seem like they're picking these up from other pathogens, maybe in a wound or in a hospital setting, something like that, where they would be kind of in the same environment with these other things that infect people.
Has anyone tried targeting the sword itself and taking away the bacteria's ability to pick up genes?
So that's a good question. The sword actually is not required for picking up genes. Remember, I said if I take it away, some of them still pick up some genes from the other species, wherever it is, just because at some slow rate it will die and release DNA in the environment, and these guys pick it up. It was just that slow rate seems to be too slow to explain how fast they were getting resistance. And I think the sword mechanism kind of speeds it up because you release DNA from the guy right next to you. It's right there to pick up. And so could we try to target the sword mechanism? Maybe that would be a good idea, like in an infection. So if you have a patient who's infected by several different species that have maybe different antibiotic resistance profiles, maybe you could try to block it and see if that would slow down the spread of resistance. But the reality is we don't really understand the dynamics of what's happening inside of the infection well enough to say whether that's safe or not. Like, maybe these guys with the swords are actually holding some other guy that's even worse in check. So it's a good idea, but yeah, everything's complicated and we definitely have to make sure we understand it more. Question?
So the things that scared me the most when I was reading about antibiotic resistance is that companies are losing interest in doing research and finding new antibiotics because they spent 20 years getting the drug on the market, and then 2 years later the drug doesn't work anymore. So that's the part that scared me. Oh, are we going to go back in 200 years where I have a scratch, I'm going to die because there are no more antibiotics that works. So do you think if there is any interest in the companies to look to that side? Do you feel, which is going to be great for you because then you can become the Duke pretty quickly, but do you sense any interest in any of the companies, like the...
So I think targeting something like this, the poison tip dagger thing, and the interactions between bacteria would. So I think one thing about companies is that they like things that are kind of proven and are safe. And yeah, developing a new thing takes a lot of money, and this would be such a paradigm shift that I'm sure it would be difficult to get them to sort of go in that direction. And then the other thing is, if you're an infected patient, you really don't care about the spread of resistance. You just want to get rid of what's in you really fast. And like preventing spread of resistance in what's in you is good for everybody else. But for that one patient, you want something that will kill everything really quick, and that's antibiotics. As for the sort of economics of why we're falling behind these cells and these bacteria in developing new antibiotics, it's an interesting economics issue that I don't fully understand, but apparently the return on investment for antibiotics is just not as high as specialty drugs. So one argument I've heard is that you take antibiotics for a few days and you're cured, whereas if it's like a heart, high blood pressure thing, then you're just on that forever. So you have a more steady stream of income. But that doesn't really make that much sense to me because a lot of people get infections and antibiotics are used widely. So. It is.
The problem mainly is that you don't have much of a window where you can sell a lot of antibiotics if the resistance spreads so
the
fast.
resistance spreads.
Right? Penicillin resistance came out like a couple of years after penicillin was invented and spread, and they thought that would cure everything, right? So if you and it takes so long to put a new drug on the market and so much money.
Yeah.
then either then you make that drug cost like $1,000 per pill so that you can return your investment, or it doesn't make any sense because you don't know how long you're going to be able to sell the drug. So to me that makes sense, but it's scary.
So there's two things. One is there's actually a lot of scientists in like academic research labs that are working on this problem because it's interesting, and they can find something interesting and not necessarily come up with a good drug, and it's still a result for them. And people have been developing new ways of making antibiotics and discovering new antibiotics in sort of more clever ways. And then in addition to that, I think the White House recently announced a big initiative for funding research into basic antibiotic resistance mechanisms and drug development, drug discovery. So hopefully that will make some difference, because I mean, it's not that we can't beat these bacteria, it's just that we got to put the time and the effort and the resources into it. So hopefully shining light on the problem, like once we realize that this is an issue, we can start to address it and catch up and put these guys back under.
Well, thank you so much. Okay. All right. Thank you. This was really fun. I really liked it. So I guess this is the end of our day 2, Brewing Company. Thank you so much for everybody for coming here. I think some people are getting some kind of little giveaway and prizes because they asked the first questions or something like that. So if you find pins or fake tattoos or posters, please take them home with you. If you want pint glasses, you can get them at the end with Caroline. Thank you so much, Jolla Brewing Company, for hosting tonight. Thank you for the 3 amazing speakers for being here. I've learned a lot, and I'm a scientist, and I like to learn about all the other science. Sometimes we're too focused on what we do, and it's actually quite nice to get outside that box and learn about something else and get scared about something else. Like antibiotic resistance. I'm really panicking. But anyway, so I mentioned, well, thank you to our sponsors, and I mentioned our collaborator, the Science Center. So if you like these type of events, this Pint of Science Festival happens once a year only. For 3 days, so it's pretty big, but only once a year. So if you want to go and see more of these type of events, go and check out the Science Center website. They have an event called 2 Scientists Walk into a Bar, which sounds like the beginning of a joke. So I think they have an event on June 2nd from 6:30 to 8:30. They are actually going to be in 25 bars. Like, I thought I had a problem like organizing in 3 different bars, but they're going to have 25 bars all over San Diego County. So they're going to be like, of course, 50 scientists all over. So I hope I'll jump in. I want to be one of the scientists walking to a bar. The cost is free. Like it. Yay! We have another event called Crossover, where let's start over. Crossover, where comics and science meet. Teenage Mutant Ninja Turtle, Godzilla, and the Science of Mutation. That sounds pretty cool. The cost is $15. You can find all the information on, it's called actually rhfleet.org. And then the other one that I know, it's Subs and Science. It's going to be on June 6th from 6:30 to 8 p.m. They're going to talk about autism spectrum disorder. So if you enjoyed the talk today, you want to know more about it, you can go there. The location is to be announced, and the cost is only $5. Okay, I think organ announcement over. Thank you. Check out our next event tomorrow here at Jolla Brewing Company. We're gonna have stem cells. I work with stem cells, so I'm really invested in that event. But there are gonna be other events also in North Park and downtown, so check those out. If you don't have reserved your tickets, walk-ins, there is a high chance that people don't show up because it's free. They reserve tickets and then who cares? They don't lose any money. So just walk in and it's gonna be fine. I think that's it. Thank you so much, everybody, and have a great time.